Hello, everybody. A warm welcome to all the participants of today's national webinar, that is Open Source Tools for Computer-Aided Drug Design and Their Applications. On behalf of Chebrolo Hanumaya Institute of Pharmaceutical Sciences, I wholeheartedly invite the speaker, allied personalities, and all the participants who have attended this webinar. This webinar is being organized by the Department of Pharmaceutical Chemistry, the Internal Quality Assurance Cell, and the Institutional Innovation Council of CHIPS in association with Indian Pharmaceutical Association Education Division. Before we start the proceedings of this webinar, I would like to present to the participants a small audiovisual about our institute, that is Chebrolo Hanumaya Institute of Pharmaceutical Sciences. That was a very short audiovisual about our institute, Chebrolo Hanumaya Institute of Pharmaceutical Sciences. Now let us dive into this webinar. Now I request the head of the Department of Pharmaceutical Chemistry, Dr. J. Subbarao, to please give the welcome note of this webinar.
Thank you, Kalyan. Today's topic is the open source tools for computer-aided drug designing and their applications. Computers are mainly essential tools in modern chemistry and are important in both drug discovery and the development of drugs. And it is used in pharmaceutical industry to discover, design, optimize new effective and safe drugs. So it is an innovative process in finding a modern chemistry and are important in both drug discovery of the new medications which are based on biological target. So it involves in designing of new molecular shapes, sizes, and the charge to fixed biological target to which they interact and they will bind. Computer-aided drug designing is an effective, efficient mode of technology for identifying potential lead compounds and for the development of possible drugs for a wide range of diseases. Today, a number of computational approaches are being used to identify potential molecules from huge compound libraries. It helps scientists, researchers in minimizing synthetic and biological testing efforts by focusing only on promising lead compounds. So it's a great time today that Dr. Bharat Kumar is going to share a valuable information about the open source tools for computer aided drug designing and their applications through this webinar from our Chebrol Anmaya Institute of Pharmaceutical Sciences. Thank you, sir. Once again, I cordially inviting you for this webinar. Thank you.
Thank you, sir.
Thank you, sir. Thank you for your welcome note. Now, I take a lot of pleasure in inviting the speaker for today's session, Dr. Bharat Kumar Inturi. He is currently working as drug discovery consultant at JC Biosciences, United States of America, with a total of more than 8 years experience in computer-aided drug designing. And his education qualification is he completed his bachelor's in pharmacy from KLR Pharmacy College, Kakatiya University, completed his master's in pharmaceutical chemistry from Manipal College of Pharmaceutical Sciences, Manipal University, and then had his PhD from JSS College of Pharmacy, JSS University. He has also received an SRF grant from Department of Biotechnology for more than 3 years, and he has published 19 research articles in international peer-reviewed journals. And he is also currently a reviewer for Chemistry Select, Journal of Molecular Structure and Futures in Medicinal Chemistry. So with this varied background and this diverse applications that he has in the field of computer-aided drug designing, we found that he is the suitable candidate to deliver this talk on this topic. And I once again cordially welcome him and request him to take over the platform and deliver his speech. Over to you.
Thank you, Raghav. Thank you, Subara sir, for your generous welcome and briefing about me. And thank you, Chips, for giving me this opportunity. Okay. Let me share my screen. Yeah. So is it visible my screen?
Yes, it is.
Okay. Yeah. So today's my topic is open source tools for CAD and their applications. Why I have chosen this topic? So the reason is when I was working as an assistant professor or somewhere, I observed that many of our colleagues are students and many other faculty members. They were not aware about what are the tools available in the computer-aided drug design. And many people thought that docking computer molecular modeling means either highly paying software like coding the Sibyl or Gold, that kind of software. But there are n number of hundreds of softwares are available, which is freely either for academic uses or completely even for the commercial purpose. So with this webinar, I'm going to give you some brief idea about what are the tools available or for the people who are looking to start their molecular modeling studies, or they want to already they are in this field, or some people like faculty or researchers already who are doing their research in the maybe synthetic medicinal chemistry, but as a part of paper publication or a part of thesis, they want to add some kind of modeling studies to their work. So with this my webinar, I wanted to give you a brief overview of the various tools. See, to learn all these tools, it will take at least 1 week of time. But in this 45 minutes or 1.5 hours of time, so I'm giving you just brief idea about the various tools. So after this webinar, maybe you can go and explore yourself. So With this overview of the webinar, I'm going to majorly talk on first. I'll give a little intro about the drug discovery, then overview or intro about the CAD. Then I will discuss little more on the molecular docking. Then I will discuss various CAD tools which can be implemented for our research purpose. So as we know that is a traditional life cycle. So like earlier in the olden days, we never had any kind of rational approach for the drug discovery. We have seen that many drugs are serendipitously discovered. So like we used to have a natural products or synthetic compounds directly, we can take it for the preclinical studies or the clinical studies in the against various diseases. Maybe if it is everything goes well, maybe we'll get a one drug. That is how we have discovered many of the drugs. But it is lacking the rationality in this. So that is where we are going to implement or going to talk about the what kind of rationality we can be put into the drug discovery process. So we know that drug discovery process involves billions of dollars of money. So to reduce this cost, can we able to implement some kind of technology? And also it takes 13 to 15 years or 10 to more than 15 years of time to discover one drug. So to shorten that process and reduce that kind of huge investment, can we implement some kind of technology? Yeah, that is where our computational tools came into the picture. First, when you start, where discovery starts, identifying the disease. So once we have the disease, so what we do, like recently we have seen last 2 years a huge pandemic. Once everyone talks of the pandemic, initially everyone did all the research, they have identified the genomic sequence. From the sequence, it started the discovery, right? So once we have the genomics and proteomics, from where we have seen personalized drug targets, like if you, so there are many companies in the United States or somewhere in the Europe, so even in India. So if you give like your DNA sample, they will analyze your sequence, and they can run through various genomic studies, and they can able to tell you in future you will be getting any kind of cancer diseases or diabetic related things. Or you can see in the fetal medicine, like in the fetal, like when it's 12 week or for 13th week of time, they will take the mother sample, and they can run for this some kind of DNA test, and from there they can able to identify any kind of genetic disorders, baby will get it. Yeah, that kind of studies already been in there. So that kind of, it all involves some kind of computational process. And next step of the technology, where we're talking about the high throughput screening. Actually, this high throughput screening, it's a whole physical process. There is no much prediction involved in this, but only thing it is doing is in the, like when you look into the big companies like Pfizer or Novartis or some kind of GSK, they'll be having their in-house libraries of molecules, maybe millions of hundreds of thousands of molecules with them. So whenever we get some kind of a target with them, so what they do, they will, by using the robotic machines, they will screen all the compounds against the target. From the target, they'll be getting one maybe a hit or lead compounds. That is where the hypothesis screening is involved. So recently, I have been working with one of the clients. So they are implementing the hybrid approach of the HDS. See, they have the 100,000 or 500,000 of the molecules. So screening those 500,000 HDS process involves a huge amount of the money. So what those people are trying to do is, first, they will be having those 500 physical compounds molecule structure as a virtual library. So they are implementing some kind of virtual screening. So they know the target. So they are doing some kind of molecular docking studies initially. From the docking studies, they'll be taking shortlisting maybe of 5,000 to 10,000 compounds. So from the 10,000 compounds, maybe instead of screening, doing HTS 500,000 compounds, here we have shortlisted only 10,000 compounds. So there will reduce a lot of our manpower and also time and also the money. So the screening of 500,000 compounds, hardly it costs us $1,000 or $2,000. It doesn't matter for the companies. So that kind of technology can be implemented. So next one is virtual screening. The virtual screening, I'll be talking more little in detail in the later session. So virtual screening involves various parameters. So you will be having a library of compounds, virtual library of the compounds with you. Maybe all we know that rule of five, that is the one of the basic parameter every researcher nowadays we use it, or some kind of molecular docking or molecular dynamics or some kind of pharmacophore modeling. So we can implement any kind of modeling over there. So we can shortlist. Screening means just we're shortlisting the compounds. Maybe we know that how much chemical space we have. We have like billions and billions of molecules with us in virtual molecules. So screening all the billions of molecules or millions of molecules, it is not at all possible. Our computational power won't support it all. So by using some kind of screening method, just we are shortlisting the molecules. That is where we can talk about the virtual one. Like then we're coming to the CAD. It's where we are going to use some kind of computer-aided drug design methods like molecular docking, molecular dynamics, QSR, pharmacophore modeling. So using all those methods, we can able to shortlist some compounds. And finally, yeah. So where we can use in vitro in silico ADMET models. So like we have shortlisted the molecules from the various studies and everything is perfectly good. But if molecule fails in the preclinical or clinical testing, then all our efforts will be waste, right? So that's the reason we can able to predict some kind of in silico ADMET these things. So far we can able to implement various technologies in this picture, but once it reaches to clinical testing, nothing can be done. It has to go through the clinical phase, all phase 1, phase 2, phase 3, phase 4, all those things. So this kind of technology can be implemented in the drug discovery process. Yeah, now coming to the latest feature, everyone talks about the artificial intelligence and machine learning. So machine learning, it's not new to the computer drug design. We have seen that like QSR studies. It is a mathematical relationship between the physicochemical properties and the biological activity. So similar kind of approach can be used in the machine learning. In artificial intelligence, it's a little new idea. We have seen so many startups are coming into the Western world. So they are doing like huge studies in the artificial intelligence and machine learning. They are getting some kind of hits. But as a pharmaceutical chemist or Indian, like who are doing like our researchers, we're far behind, like 1,000 miles behind to learn the artificial intelligence and machine learning. So with this, just I'm moving to the CAD. So what kind of CAD technologies can be used, like based upon the information available? For example, if you already knew the protein structure, when I'm talking holistic structure means it's a protein structure. It's ligand means it's a molecule. Okay. When already we knew that some kind of protein structure and also known ligand both are available, then we can go ahead with the structure-based drug design approach. But in another scenario, we knew that there is no ligand is available. Only the one protein structure is available. In that case, we can take it for the de novo design. And some other cases, we'll be having some kind of ligands, but we don't know where those ligands are acting. We don't know the target, but we're getting some kind of response to the disease. So on those cases, maybe we can take up the ligand-based drug design approach, like similarity searching, pharmacophore like modeling, or QSR. This kind of things can be done. If there is no information at all, either we don't have ligand, we don't have anything, that is, we don't use any kind of CAD. So we need to start from the scratch. So as from the above slide, we can able to see there's majorly two types of drug design strategies. One is ligand-based drug design strategy. Another one is the structure-based drug design strategy. Ligand-based drug design strategy, where we do not have the protein structure or the target of the disease. Give me a second. Where we do not have the protein structure or target of the structure, then we can take up for the, but only few ligands or the molecules are available. In those cases, we can take up a ligand-based drug design approaches. Like maybe we can talk about the QSR, or we can go ahead with the pharmacophore modeling, or we can go ahead with this similarity search. Those kind of things can be used in the ligand-based. Structure-based drug design approach, where we have the protein structure, three-dimensional protein structure with us. Maybe we knew the target site or binding site. Then we can go ahead with the virtual screening or molecular docking or molecular dynamics. Those kind of things can be done in these cases. So if you look at the if you look at the whole picture of the computer-aided drug design, how can I hide this episode? Is there any way? Okay, leave. So if you look at the, is there any way to hide this above bar?
I don't think so. The black one?
Okay, I got it. Let's hide. Yeah, okay. So when you look at the whole picture of the CAD, we have structure-based drug design approach, where we are having the three-dimensional structure of the protein. Then from the three-dimensional structure, we need to identify the binding site. So when you have the like 300 amino acid sequence or 500 amino acid sequence of the protein, so we'll not be using that whole amino acid sequence for our drug discovery process or our molecular docking process. Only from the protein, we'll be taking some part of the structure. We can talk it as maybe a cavity or the binding site. So out of those 300 amino acids, we need to identify few amino acids which are responsible for the binding site, like interact with our ligand. That we can talk it as a binding site. Once we have the binding site, then we can go ahead with the docking and scoring. Or else we have a few ligands with us. We do not have any protein structure. In those cases, we need to take, like for example, if you have a 20 plus molecules with you, and along with the biological activity, maybe IC50 values, then we can go ahead with the QSR. QSR is the mathematical relationship between the biological activity and the physical-chemical properties. So based upon that, we can generate a two-dimensional, three-dimensional QSR models like COMFA, COMSIA. Those studies can be done. Or else you do not have any biological activity, or else only hardly you will be having one molecule or two molecules, 5 molecules. So with those 5 molecules, just we'll be identifying the common features of the molecules. Like what can be the common features? Maybe they are sharing all same aromatic rings, or maybe electrostatic groups or hydrophobic groups, or some kind of rings. So based upon that, we'll be identifying some features along with the distances between those features. Then using that pharmacophore model, we can search against the various databases. So with that databases, we'll be getting the similar compounds, not similar compound in the terms of the property sharing. So the hits we are identifying, those hits will be having the similar properties, like those will be having same kind of aromatic ring, maybe a hydrophobic groups or electrophilic groups, those kind of things. With that, we are having some kind of ligands. For example, if you have a 500,000 like virtual library of molecules, by using these techniques, you can take it for the virtual short. You can shortlist the maybe 500 molecules or 1,000 molecules or maybe 100 molecules. So from this, maybe you can take it for the molecular dynamic studies from this, or else right away you can take it, you can shortlist the compounds and you can take it for the your synthesis. Or else here you can do it the in vitro ADME studies. So after that, you can select the compounds, then you can take it for the synthesis, then you need to identify the leads. From the leads, maybe you can take it for the preclinical, then clinical, finally you'll get it drug. This is the whole picture of the where that we can use the CAD. So I'll be giving little intro about the ligand-based drug design. So here, this is a ciprofloxacin. If you look at this structure, it is having some kind of hydrogen bond donor groups and acceptor groups, and also it is a sp2 hybridized ring here, and also we are having some kind of lipophilic parameters, and also we are having some kind of here size. Here you can see 3-membered ring is here. It is here some other ring, 6-membered ring is here. So, and also it is the charged positive groups in this position, and it is the electropositive groups who are here. So from this one molecule, we're able to get this so much information. So based upon this information, maybe we can get some knowledge, but it relies on the knowledge or other molecules that bind to the biological interest of the targets. So these properties are responsible some kind of interaction at the target site. Maybe these properties are very much essential for the activity. So with this, using these properties, maybe we can derive a pharmacophore model. So that defines a minimum necessary structural characteristics a molecule that possesses in order to bind a target. So with this pharmacophore model, we can search against some library. So maybe we are going to talk about the libraries little later in this session. Okay. A model of the biological target may build based upon the knowledge of what binds to it. And the model, the terms may be used to design a new molecular entities. So once we have the model, we can search against various libraries. But from that libraries, we can shortlist the molecules. And alternatively, we can take up, for example, as earlier I said, if we have the 20 molecules to carry out the QSF studies, at least we should have a 20 molecules. From the 20 molecules, we need to divide those 20 molecules into test and planning set. Then from that, we can generate some kind of mathematical model like COMFA, COMSAT, 2D QSR, HQSR, those kind of things. From that, we can predict the activity of the new molecules. But as of now, nowadays, most of people not using the QSR when they are behind the molecular docking or molecular dynamics, some kind of pharmacophore search, that kind of studies they're doing. So that's a little about the ligand-based approaches. Then we're moving into the structure-based drug design strategy. So when we are talking about the structures, from where we'll get the structures and how we can determine the structures. So the three-dimensional structures can be derived or can be discovered or identified, obtained through the various approaches. One of the major approaches is X-ray crystallography. So what are the PDB structures today, we have 90 to 95% of the structures are derived from the X-ray crystallography. And for X-ray crystallography, the minimum need is the molecule should get at least one single crystal. Then from that we do X-ray diffraction. From that we'll be making the electron density mapping. From that we'll be getting three-dimensional structure of the protein. Or in some cases, there is a crystallization is not possible. So for all the molecules, it may not crystallize, especially we can take some GPCR or some other proteins. Crystallization is not possible at all. In those cases, we can take it as NMR. But the problem with NMR, it is a very complex process. We know that, but elucidating very small molecule for us, it takes like days and weeks of time. So just think about like 20 atoms or 30 atoms of a synthetic molecule. It is a very difficult to elucidate it. But when you talk about the 300 atoms or 500 atoms or 1000 atoms protein structure, how difficult that could be with NMR. And next technique nowadays is electron cryomicroscopy. So based upon the three-dimensional data, we get it from the cryon microscopic, we can obtain the protein structure. Or else we can combining the multiple methods, above methods, we can get the three-dimensional structure of the protein. So, but what are the source for the getting a three-dimensional protein? We should have from where we'll get it, either from the Homo sapiens or from the plants or from the bacteria, virus, fungi, or DNA, RNA, anything can be a source. From the source, by using the like above techniques, first we need to express the gene after the gene expression, we need to cultivate the gene. From the cultivation, we need to purify that protein, then we need to take it for the crystallization. Once you have the crystal structure, by using the above X-ray crystallography method, maybe we'll get a three-dimensional protein. Once we have the three-dimensional protein, so there it is a very useful like web resource we have. It's a protein data bank. So most of the people who are derive with the three-dimensional protein structure, they deposit. It's not a mandatory deposit into the PDF data bank, but they generously people deposit into the protein data bank. It is one of the open source tools. From there, everyone, all like us, we can use it for our discovery purpose. And if you look at the PDB database statistics, so it was all around 1995. It started long back. It started, but if you look at the recent statistics, as of now today we have approximately 180,000 plus structures we have. If you look at the last year statistics, around 14,000 new proteins were added into the database. Here you can see the dark blue color is the every year adding structures. And maybe see if you look at the last year's number, it is looks little higher, maybe due to the recent pandemic. So yeah, we knew that we have a protein structure with us, but how do we choose that protein from the protein data bank? That I'll be talking little later when I go into the like demonstration about it. Yeah, first once we have the protein structure with us, we have selected from the PDB data bank. So then we need to prepare. Straight away, we cannot use that structure for our molecular docking or dynamic or modeling studies, because the X-ray crystallography cannot add hydrogen bonds to the protein. Why? Because the hydrogen is a single electron atom, and the electron is little far away from the center nucleus. So the X-ray crystallography cannot able to identify correct order of the hydrogen bond. That's the reason it won't add. So we need to add hydrogen atoms or missing hydrogen atoms to the PDB structure, and also like it contains the like when they're doing the crystallography, it will have some kind of solvent or some kind of salts they add into it, or it will have the water molecules. So in general, when you're talking about the molecular docking, so we usually remove all the water molecules from the docking study, except if you know we are very much sure about any specific water molecule is very play a crucial role in the molecular docking study. As of now, I never kept any kind of water molecule into the molecular docking study. So by not blindly, most of the 99% of the time, you just delete all the water molecules from the protein. And also sometimes when they are doing a crystallization, so if more than one sequence get crystallized, if we can talk about chain A, chain B, chain C, chain D. So it will be having multiple chains in that protein. So in those cases, we need to choose, first we need to analyze the sequence. Is there any missing amino acid residues in that sequence? Then we should not choose. Or we need to choose a best available sequence. For example, in sequence A, 2 amino acids are missing. In sequence B, 20 amino acids missing. So in those cases, sequence A is better than the sequence B. And in next scenario, while crystallizing the protein, most of the times they will add one ligand to the protein. So where we're calling it as a co-crystallized ligand. So in the co-crystallized ligand, for example, chain A contains the co-crystallized ligand, chain B doesn't have a co-crystallized ligand. So always we should choose the molecule which is the protein which is having the co-crystallized ligand, because the co-crystallized ligand will be very valuable information that decides where our binding site is. So as earlier I said that out of 300 amino acid residues, it will have the multiple cavities. Out of the multiple cavities, maybe one cavity is responsible for a binding site, where our molecule or ligand will attach, our co-crystallized ligand will bind to it and it will give a desired activity. So from that, we'll be deciding the binding site for our study. And also it will have the protein side chains. Maybe we need to remove the side chains, and it gives the different electron density. It will be having isoelectric groups. Just we need to correct all of them. And in some cases, effects like aspartame, glutamine, histidine. So the positions of the hydrogen bond, we need to correct it, because maybe these, like for example, this flipping, just look at these flippings. So here we have NH2 in this side, this is this side. So if you flip it wrongly, you may miss the hydrogen bond with your ligand. So in those cases, you just need to be little worried. Maybe most of the cases, the software will take care of it. But in manually, you also need to take a look into this. And once you have protein will be ready. So in the protein preparation, first you are adding the complete hydrogens to it. And if it is having the multiple chains, you are deleting any of the chain. That is good. After that, you are removing the complete all the water molecules. Then you are choosing the target site. Based upon that, you are preparing the protein. Once protein preparation is ready, then next part, ligand preparation. So ligands. So when you are working, like you are synthesizing some molecules, you are about to start some project, maybe a master's thesis. So initially, you'll be having some idea about some synthetic scheme. From this, you'll be having maybe let's just take a common scenario. So like we usually synthesize molecules based upon our chemical availability in our lab. Maybe we'll be having like 10 aldehydes and 10 amides. Maybe using that together, we can synthesize various molecules. So already we know that what kind of final out molecules will come out. So instead of using 10 aldehydes and 10 amides, so it will be generating at least 50 to 100 molecules. So is synthesizing those 100 molecules is it worth it? We don't know, right? So that's where the rationality comes in. So first you draw, you design a synthetic scheme. From that, you decide how many derivatives you can able to generate it. Maybe 100 molecules, example. So from the 100 molecules, we can call it as a virtual library. Just draw all the 100 molecules in your computer by using any kind of ChemDraw. ChemDraw is not a free software. That's where I'm going to take another tool. It's a Marvin Sketch. It is a Marvin Sketch is a academically free software, or ChemSketch. It's a free for academic. So the reason why I'm not suggesting you can use it in ChemDraw, but when you're going for the publication point of view, so you are unauthorized to use ChemDraw. It is illegal to use ChemDraw because we do not have any license for that. That is where I'm talking about open source tools. So you can draw a structure, then you need to convert into three-dimensional. Maybe you have the converting three-dimensional. You can use the Marvin Sketch or OpenBubble. So those kind of structures. But whenever you are drawing structures, just you should know the file format. File format is something like we have the Word document. The extension will be DOC. You have a PDF document. So whatever the information it has, the same information we have. It is either PDF or the Word, but only the extensions are different. Similarly, in the chemical files also will be having various file formats. It's a SMILE or MOL, MOL2 or SLN or STF. So SMILE, it's a very simple format. It will be having only sequence of the atoms, and it will not contain any kind of hydrogen. So MOL format, it will be having 3D coordinates of the various atoms in the file, along with the we can add some kind of properties. Like in the MOL file, we can add the number of hydrogen bond acceptors or number of hydrogen bond donors. Those kind of all kind of information can be added. Maybe later I will show you one MOL file, so you'll get clear idea. So in the difference between MOL and SDF, like MOL file will be having only one structure, and the hundreds of MOL files can be clubbed together into the SDF files. So if you want to use like 100,000 molecule ligands for the study, so your system may not handle, for example, it is a small system like 2 GB RAM and 2 CPUs. Like it cannot process 100,000 files in your laptop. So in this case, in those cases, you can use one single file like SDF file. Once you have the three-dimensional structures of the molecules, maybe then you can take it for the minimized structure. So the minimization of the ligand is very much essential because we know that the least energy state molecule is stable. Once we have the minimized structure, maybe you need to convert into the required format, and each and every software may need a different file format. Maybe some softwares need a MOL format, some require a SDF format. Most will accept the SDF. Maybe some other, like for example, if you are using Autodocs, in that file, it needs a PDB-QT file for the molecular docking. So maybe we can use, like I'll be talking later in this session, what kind of software we can use to generate maybe this kind of files. Okay, this is all about the protein preparation and the ligand preparation. In the ligand preparation, just to draw a structure, then convert it to 3D, then minimize the structure, then save it according to your need, based upon the software needs. Okay, then protein ligand docking. So here, just we have the protein with you, and based on the protein, you just need to generate the binding site. Then you have the docking. So you have the ligand with you. Then we need to it will get together a program, a molecular docking program. Dock into it. It will generate a maybe a complex. So the complex predicts the pose of the molecule on the binding site, and also it generates a binding affinity, how much, and also the interactions at the target site. So the pose versus the binding site. So pose is the molecule, how exactly it binds at the. So binding site is the cavity in the protein structure. Pose is the ligand, how our binding mode of the ligand at the binds at the binding site. So how will it like shortlist molecules? Like when you have 100 molecules of a doctorate, and so you need to consider especially main three parameters. Always docking score. It's not the main key parameter. You need to consider docking score and the hydrogen bond interactions and the pose. So these are the three parameters very much essential, because like if molecule is very big, definitely it will give better score, but it may not have the required pose. So how can you come to know which is the very good pose? By comparing your molecule with the co-crystallized ligand. If you have any co-crystallized ligand or any known molecules with you, so based upon it, just you compare both the molecules. So my molecule is making similar kind of pose at the target site, then it's good. So our score is good, pose is good. Then next one is interactions. So in every case, there will be some essential interactions or catalytic interactions that are very much essential to generate the activity, right? So if my molecule is making that kind of interactions, then it is satisfies all the three parameters. One is it is having the good docking score, and it is having the good docking pose, and it is generating good interactions at the target site. Then you can shortlist that molecule. Then so what is that's about the little about the docking. Then we'll be talking about the uses. So how what are the uses of the docking? Mainly the post prediction and the virtual screening. If you have the hundred thousands of molecules, you cannot synthesize or you cannot purchase those hundred thousands of molecules. Just do virtual screening, use molecular docking program. You will be shortlisting 100, 500 or 1,000 molecules. Then also, or else you have one molecule, maybe you have one natural product, like you have taken one natural plant and you have elucidated a structure from the natural that plant, but you just wanted to know, like, is that molecule is active against any kind of target. Maybe you can take it for the various cancer targets, tuberculosis targets, and you can verify how that molecule is binding at the target site. From that, you can able to identify the pose of the molecule and what kind of interactions it is making. So just remember that there is no criteria for docking score. This much need to be there, like -2 should be there, or -10 should be there, -20 should be there. There is no specific criteria. It changes from the scenario to scenario. From one target, it will be -4 is the best one. Another one +6, -6 is the best one. Another target, 10 is the best. So always just you keep it as a reference, maybe based upon your co-crystallized ligand. The reference ligand. So your molecule binding score should be better than your reference ligand. That is the only criteria you have. And that is all about the docking program little. And when you go deeper into the docking program, so we never need this thing, what kind of algorithm it uses, we never thought about all these things. What kind of program it uses? It is called a search algorithm, and what kind of scoring function it uses. Just what we do is directly we download the software, straight away we run it, right? So, but usually any docking software, it contains two basic parameters. One is search algorithm, another one is scoring function. Search algorithm is something like how it searches. For example, if we take the molecule, we know that molecule can able to generate a large number of confirmations. How can it choose out of those confirmations? How can it able to search all those confirmations against the target? And how can it shortlist? Maybe next comes into the scoring function. So we have 100,000 confirmations for one molecule. So out of that, how can it choose? Maybe, for example, in the docking program, we can able to choose 10 poses, 20 poses. So out of those 100,000 confirmations, how it is choosing only those 20 poses, maybe based upon the scoring function. The pose of the molecule in the binding site, it gives how it can able to fit into the target site. Binding affinity, it correlates with the score of the molecule. And we have seen various parameters in the docking. So what are the available softwares we have? There are hundreds of software. We know that Schrödinger is one of the highly commercial software. It is not free, like it is highly expensive. After that, we have a Sibel was there, and Discovery Studio we have, and like free software. These are most of the some of the free softwares we have. Like Autodoc is one of the most popular free software. Another one is the little expensive. It was costing hardly maybe $1,000 per annum or something like that. When compared to like other commercial software, Gold is a much cheaper one. So if you have a little funding with you, you can go for Gold, or you don't need to purchase any software. Straight away, you can use Autodoc or some other doc or Heats or some kind of thing you can be used. So each software will be having different scoring functions and different search algorithms and different approaches. Okay, but yeah, they might have done lots of research on that. So maybe we can choose any of the program and we can proceed it. Okay, only thing you should have some kind of rationality. Why you have chosen what are your molecules and what are your targets. So in those cases, you must have a rationality. Then we can go ahead with any kind of program. Yeah, set space. So we're not talking about the protein and ligand in a part, but it involves straight away. It cannot go and bind at the target ligand. There are many degrees of freedom it involves. So the translation and rotation of the one of the molecule related to the another involves 6 degrees of freedom. Right? Then along with 6 degrees, and also we know that there's a conformational degrees of the freedom, both ligand and the protein. So when a ligand and a protein joins together, what will happen? There is an energy liberates. There is an energy liberates. Then there is a conformational change in the maybe a ligand and also the protein. So definitely the whole scenario will change. And also we know that 70% or 80%, most of the human body or anything, it contains so much solvent. So the solvent also plays a role in that. But when we are considering a molecular docking program, we usually ignore the solvent effect. If you want a solvent effect, you want a more flexibility in the protein as well as the ligand, we can go ahead with the molecular dynamics. Where it involves the solvent effect and also momentum will be there, and we can maintain this constant pressure and temperature. So those kind of parameters can be chosen. So compared to the molecular docking program, molecular dynamics, where we can get the binding free energy that is more accurate. So if you want to compare your results activity, biological activity results with the any of the docking or dynamic results. So you always prefer to compare with the molecular dynamic results, so that will be more accurate. So the search algorithm generates poses, orientation, particular conformation of the molecule in the binding site. So search algorithms, yeah. So it will be having a rigid docking and the flexible docking. Rigid docking, where ligand treated as a rigid structure, there is no movement at all. That directly it will, what are the pose it will getting, what are the conformation we are having, directly it goes to the target site and it try to find out the best pose, and it will give the result. In the flexible, the ligand is flexible. There is various conformations will be generated. In the rigid docking, you just need to generate conformations yourself, right? So, for example, Freed, fast rigid exhaustive docking or OpenEye. So, the rigid docking is much faster but least accurate one. Coming to the flexible docking, it is the most common docking study. Like conformations will be generated on the fly. So on the fly means once a molecule reaches to the target site, there is based upon the dock program, it generates the number of conformations. Okay, based upon the energy, it will calculate the energy at the target site and where it reaches to the least energy of the conformation. Maybe from there it stops it. So from there it will try to identify the best active molecule, best pose. So then it gives the like best required poses, and it is having the two kinds of approaches. One is stochastic method. So in the stochastic methods, like we'll be using genetic algorithm for the, like, especially if you talk about the gold software, it is the genetic algorithm, or it is a Autodock, it is a simulated annealing. So simulated annealing is the temperature of the like conformation. It generates increases gradually. And also another one is incremental construction. It is a series of stages. So, but for this, okay, one issue with the stochastic search is there is no like rationality being generated in the conformation. So it won't give the optimal solution, but reasonable length of the time. So it generates some conformation and it try to identify the best one. So here we'll be reducing the time for the docking. So for example, we have the like 1 million 10 lakh compounds. From that, if you want to choose a it's been 12 o'clock. Okay. If you want to choose some molecules, okay, you can go for stochastic. Incremental construction, it takes a lot of time. So maybe but it will be having more accurate one. So in those cases, it's a flex dock, like Surflex, those kind of docking program will use it. So when we talk about the ligands and all, we know that Charan, both conformations, which is having least energy, that is a stable one. For example, so how many conformations for each molecule will generate? For example, if we have a torsional or rotatable bond, so if each torsional angle is rotating 30 degrees together, how much it can able to rotate? Maybe 360 by 30 degrees. It will give 12, right? So if you have, for example, 6 torsion bonds with you, it will give you 12 to the power of 6. Just can you imagine how many conformations it will generate? It will generate 29 lakh conformations for one molecule, which is having the 6 torsions, right? Just imagine this like tax one, how many conformations it can able to generate. So, and also the ring conformations, all these effects. For example, if you look at the a molecule which is having, this is the active pose, or it is the best pose, but you can see that how many conformations it got generated at the target state, right? So out of that, this green color one is the best active conformer. See, the conformations generated from here to here is the whole space it got covered. But out of that, this is the best active conformer. Okay, so next one is protein conformation. So most of the docking softwares are the protein as rigid, unless explicitly if you allow to protein in some extent. The whole protein you cannot allow to flexible because it needs a lot of computational power. That's the reason most of the software is the protein is rigid, it won't flexible, but there will be in some programs you can able to allow some particular protein site, you can able to allow it. So In that way, you can go for protein conformations, or you can do flexible this one. And next one, perfect scoring function. So the perfect scoring function should accurately calculate the binding affinity, and actives docking score of the active molecule should be higher than the inactive molecules, and also the correct pose of the active higher than the incorrect pose of the active. For example, we have to choose in the earlier slide we have look into that this molecule can able to generate these many conformations, but you should remember that this conformer docking score should be much higher than the all these conformers. Then that is the best docking score function. So here you need to choose three parameters. One is it should calculate good binding affinity, and also it should give very good docking score for the active molecules than the inactive molecules, and it should give a good docking score for the correct pose than the incorrect pose. Yeah. Next, what are the various types of the scoring functions we have? I'm not going in depth into this. We have a based upon the force field based scoring functions, empirical scoring functions, knowledge based scoring functions. Yeah. Force field based, we have a gold dock and autodock. This will use the force field based conformations. Empirical, like PLP, glide, we have a chem score. These are the empirical based scoring functions, and based upon the knowledge, based on the statistical analysis, there are some scoring functions we have. So what are the scoring functions we have discussed? Those are the not at all. They are the not constant. I mean, not only those. Maybe we can use some kind of alternative scoring. That's consensus docking. For example, you have I have done the docking studies with Autodock. So just try to carry out 2 or 3 times. Then you just average it. That will be consensus docking. And rescoring. Just use a multiple scoring functions and do it a rescore. Okay, maybe like the consensus combined with several rescoring. Okay, based upon the various these consensus rescoring and this kind of things can be alternatively we can use it for the molecular docking study. So this is the most important thing. How can you validate a docking program? First and foremost is you should choose a molecule based upon the pose, comparing with the co-crystallized ligand and the interactions and the docking score. So the RMHT is the accurate validation procedure. So already you know that we have a co-crystallized ligand with you. It is there in the protein data bank. So it is having some kind of parameters with them. So before docking and after docking, just compare the root mean square deviation between the co-crystallized ligand of the before dock and the after dock. The value should be less than 1.5 angstroms. Then or 2 angstroms. That gives a best. That means our docking program is validated. That is one thing. And another thing. So just look into this picture. Here we have a structure of the 3 structures we have. If you look at the structure, it is a very good align. See, all the parameters. One is the gray one is the before dock, another one is the green one is the after dock. Okay. So if you look at this, it is properly aligned. But now look into second structure, this one. Here you can look at that. Yes, this part is aligned properly, this part is aligned good, but this is not 100% properly aligned. There's a little deviation here. This is also okay. But if you look into this structure, see, for this here we have a methyl group is here, for this structure, green one, and this one is here. So these 2 structures come differently aligned. This is wrong docking program. Our docking program is not validated. In this case, it's okay, it's very good. This is okay, this is wrong. So in this way, you need to look into the poses of our docking program. And also you can do another virtual screen based one. So along with your docking study, just add some known active molecules. So after the docking program, knowns should list as their actives, and knowns actives as actives should come, inactives should come as inactives. And so we need to dot all the molecules by rank each square. Ideally, all actives would be a part of the top of the list. That is another one. So next, coming to the virtual screen. So virtual screen, there is no specific criteria, any kind of parameter. You have, for example, large number of molecules with you. So you can use any kind of like criteria or parameter you can use to screen the molecules. For example, you have a you can use docking score, or you can use some kind of CDQSM method, or you can use a drug like or lead like parameter, or you can use the toxicity related parameters, or bad poses, or pain. There is pain filters. So here our target is just rank the molecules or the shortlist the molecules. Our main aim is to eliminate the some of the molecules from our study. That is all about the virtual screening. So here it's this process. You will be having general filters like drug-like filters, ADMT filters, sub-filters, based upon the actives known 3D structures. Based upon that, just you need to shortlist the molecules for based upon either for ligand-based or structure-based methods. So just like virtual screening is simply just for the shortlisting the molecules. So this is overall about the basic approaches for the molecular docking. So this is protein ligand docking is essential tool. We cannot say it's mandatory, but one of the recent days it's one of the essential tools for the computational drug design. It is widely used in the most of the pharmaceutical big companies. Now they have started using artificial intelligence, machine learning, those things, so they're doing it. And there was some success stories and many unsuccess stories we have, but it's not a golden bullet. So giving a best docking score doesn't mean that it's a very good active molecule. Always the experimental thing is the final one. And like we need to care need to be taken in each and everything, preparing protein and choosing protein, preparing ligands. So if you're doing some error, it leads to the inaccurate results. Okay. So the more information we have, the better chances. For example, if you know the target binding site with the protein, that is a very good information. If you do not know the target information, that is very bad. In those cases, it's at the end it may be ending with the least accurate results. Yeah, if you like, this is my suggestion. If you want to read very good, get a good basics of the computational drug design, just follow this book. You don't need to follow. It's a very good book for medicinal chemist. The concepts are clear, very good. Various tools. What are the tools can be used for the drug design? As I said that protein preparation. So you can use first. You can use it for the protein PDB data bank. So yeah, the protein data bank. I would say the rules to choose a PDB protein from the EVA data bank. For example, there is a one PDB protein is available. You don't have an option to choose. But for example, if you choose INH, it is one of the targets for the Mycobacterium tuberculosis. So yeah. So it has given me approximately 100 plus results. First rule is Mycobacterium tuberculosis is my target. So you need to choose a protein which is derived from your target. Here you can see we have one from Homo sapiens, another one from the Bacillus, Pseudomonas. There is multiple sources of target sources proteins are available, but you need to choose a protein from which is our target. Next one is method. So always prefer to choose a X-ray diffraction. So that is the most accurate one. Next one is the important one is the RMSD value. Always try to choose a RMSD in between 1.5 to 3. So according to some studies, the RMSD value of the protein in between 1.5 to 3, it will give a most accurate one. And also choose the proteins which are maybe latest one, last 2 years or 3 years or 5 years, the proteins are available, just choose that one. Another very much important criteria is based upon the co-crystallized ligand. If any of the protein is having a co-crystallized ligand, let me open a, okay. I'm opening one structure to NASD. Yeah, it is also INHA protein. Okay, if I look at this, it is having the A chain and the B chain. First thing is just you need to look into the sequence of the protein. So usually this protein doesn't have any missing residues. Both the chains are absolutely perfectly fine. Just you need to compare the missing residue. See, this is the PDB instance. This is the UniProt. So okay, let me show you. So where you will get the genomic sequences for the searching. So we will be having a UniProt. There is another site we have UniProt. This is the UniProt. So if you search it here, maybe by UniProt ID or maybe this is the ID. There it is. This ID, by searching that, you can get the sequence. Just compare the sequence. If anything is missing, there will be a blank space over here. So in those cases, you should not select that kind of protein. So first you need to let's compare the sequences, and the sequence should not miss any kind of amino acid residues. Then you should go for the three-dimensional protein, and you should check the ligand. Okay, this is the ligand. So this ligand should be, if you know already, you were synthesizing some ligands in your lab. So the co-crystallized ligand should be somewhat similar to your ligands. Then only you can able to compare your ligands with this ligand. Then only you can able to derive like maybe some kind of some kind of rationality you can able to show. You can tell the reviewers, my ligands are binding similar to the co-crystallized ligand. That assumes that my ligands will have the similar kind of interactions with the target site, right? So that gives some kind of idea, and also you can compare, like when you're validating it, you can tell that my ligands are binding similar, my ligands are giving better talking scores than the co-crystallized ligand. And also from here you can able to identify the interactions. Okay, this is a very good tool, you can explore it. Okay, you have the PDB if you have the PDB protein, then that's fine. If you do not have any PDB protein, what you will do? In those cases, you can go ahead with the homology modeling. So where homology modeling is something like, if you do not have the three-dimensional crystallized protein with you, so based upon the availability data, you can able to generate, or you can able to build a model. So that model may resembles three-dimensional protein model. So maybe like if you want to carry out some kind of modeling studies, you should need a three-dimensional protein that can be built by the homology modeling. So how can you do the homology modeling? From this UniProt data bank, you'll get to know the sequence. Once you have the sequence with you, then you can go ahead with the NCBI BLAST. Let me show you. Yeah. Once you have the NCBI BLAST here, just you can paste the you can go for the protein BLAST. Just you paste the sequence here. Then you choose a data bank as a protein data bank. So then just search it. It will take 2 to 5 minutes. Then it will give the results like this. What these results mean that these are the various like similar kind of proteins, maybe which is having the percentage. So what we are doing is here, just we are searching the sequence against the various other available protein structures. Maybe some protein will be having the 70%, some will be 90%, 100% similarity to the your protein, but they are not exactly as like your protein. Then you need to do some kind of modifications into that. That's where another tool came into picture. It's a modeler. It's an open source tool, and it is one of the very best tools for the molecular modeling study. Homology modeling. Just it's a little programmatic one, but nothing to worry. Very simple thing. So in the you need to run some Python scripts. If you go through this tutorial, it's a very much easier. Just first thing is just you need to paste a sequence. At the end of the sequence, the star must be there. Then once you have the sequence, just run a script. The sequence will be aligned. Once sequence is aligned, then you need to add to the after the blasting the MCBA database, you'll be getting some kind of PDB structures over here. So just open those, just collect those PDB IDs, access IDs, just paste it here. Download that and paste it here. Once this will compare all the PDB results, and it will tell you which one is the most similar to your sequence. Okay, here you can see the sequence similarity, and here you can see the RMSD value. So the good RMSD value and the highest similarity, that kind of protein, you can choose it for your modeling study. Then you need to align your model from the script. What we are going to do is just you are aligning your model with the available PDB protein. So it got aligned. Then just change the names. Just look at this tutorial and change the names. From this, align the this one protein and the sequence. It will give the various structures, which is homology model. Like where the sequence got missed, it will correct the sequence based upon the available data. It will add it to that. So at the end of this, you will be having 5, or what are the number based upon this number here. Ending model number is 5. That's the reason 5 PDB structures got generated. Okay. Once after this, you just go ahead with the model evaluation. From that, you can able to choose the best model. So this is very simple homology modeling with this tool. Yeah. Once we have the homology model with you, then you can maybe based on the predictions using the discovery this tool. This is the Discovery Studio Visualizer. This is a free tool. You can use it for any kind of purpose. So this is one of the very good tool. You can use it for the molecular modeling studies. Like if you want to edit the protein, or if you want to delete the water molecules from here. So let me open one protein. Okay, wait to MSD. Yes. Yeah, it is okay. Just to Control H, it will give some. So if you want to delete a B chain, just select on the B chain, just click on delete option. If you want another B chain here, this is a delete. You want to delete all the water molecules, delete. So that's all. All the water molecules got deleted. It is got almost it processed. But you want to see some kind of ligand interactions. Yeah, see here, it's you got the various ligand interactions with the target site, right? It's a very simple tool. You can use it for your studies freely. You can download. And another one, and then another visualization tool is Camera. This is also a good tool. You can use it for the like getting images for the papers, or you can use it for the PyMol also. That is another good tool where you can use it for the very good graphical images. You'll get it for the publications. Okay, those are the various tools we have, and where we are. Yeah. So once Yeah, another model is a Swiss model, also can be used for the homology modeling. There you just need to submit the sequence, automatically it generates various models and give it. And structure preparation, yeah, I'm not going through. Actually, I thought to give a demo on this, but yeah, it's already 12:20. Okay, so we can give a like Marvin Sketch or ChemSketch. As I said, these are the alternatives to the ChemDraw. These are not completely free, but academic purpose, these are free. Just go to their site and you can enter your email ID, just register for the non-commercial purpose. It is easy, just similar to the ChemDraw only. Just if you explore for a couple of hours, it will be much easier. So just similar to ChemDraw, okay, here itself, you can go ahead with the cleanup in 3D and you can generate the three-dimensional structures. And all those things can be done. Okay, yeah, just I want to cover very much another very much important tool. Yeah, this okay. Before going to that, I'll show you one tool. Yeah. So anyone wanted to do a molecular docking with the Autodock, just use this tool. But this tool can be done only in the Linux, or maybe you can try with Ubuntu. So what it does is, if you have a two-dimensional structure, it can able to convert them into three-dimensional, and it can able to minimize the structures using various force fields, and also it can able to generate a PDB file of PDB. So if you have the two-dimensional files, if you just draw two-dimensional SDF files, it will do whole process for you till the three-dimensional PDB files. Directly those files can be used for the molecular docking. Just maybe all these things I'm giving it is in a one Word document. I'll be sharing with you. Sorry. Yeah, this file contains all the information, what are the tools I'm talking about. It have the each and every, like along with the links to their websites. From there directly you can download and you can explore on all the tools. Another very much important tool I wanted to cover it here is Data Warrior. Maybe few of might have heard about this, many have not it. Okay, Data Warrior it's very good software, and it can able to calculate all the properties for you, and it can able to generate it can able to do a similarity search for you, and it will have hundreds and thousands of properties to generate. For example, if you want to calculate a rule of 5 parameters, it's very simple. Just calculate properties, calculate properties. What are the properties you want to choose? You have thousands of properties here. Just acceptors, donors, or drug likeness. So all the things. Just click it OK. Just matter of seconds. It can able to draw for. It is for 650 molecules. It calculated within a fraction of 2 seconds. Okay. Directly from here you can explore. Save special text file. So straight away you can explore all the molecules into all the data into text file. From there you can put it in a thesis. Or you don't want some of the few things here, just directly delete ligand efficiency. I'm not ligand efficiency. Surface affinity, delete it here. Okay, just delete. So you can delete all the details, and you can delete the structure also from here. If you want to add graphs, see, for example, here you have another column, XY. Just choose what are the parameter you want. On a side, just I want to graph between hydrogen bond donors and the ligand efficiency, or mean of value. So it's but this data, this graph is not that clear, but yeah, if you put it the proper data, this is very clear. Directly from here you can see it. So drawing graphs also very much is here, and you'll be getting lot of three-dimensional tools here. And for example, if you want to sketch the if you need a data of this molecule, if you want to do a Google search, just do right click and search Google Scholar. Straight away it takes to the Google. It is going to Google Patents. Yeah, it went to Google Patents, and it will search based upon the SMILES format, and it will get the data. Okay, so this is very good tool to explore. So as a part of your bachelor thesis or master thesis, the tool is more than enough for to generate all physical-chemical properties. And another very good tool we have, the ADMET properties calculation. So if you look at the so before going to ADMET properties, I want to show you. So in our ChemDraw, you might have drawn files into like CDX format, but you need another mole format. So converting sometimes file formats, it will be very difficult for you. So in those cases, you can use your open bubble. Just here, input file as a, for example, CDX. This is the ChemDraw binary format, or input file as a mole format. These are the various file formats. So just choose any of the file format, just here, then select your molecule, then just click it. Then from here, you choose the required format. Here, I have a STF format with me, one molecule, and another, I need a mole format. Just do a convert. Okay, this molecule got converted. Then directly, you can write the output file here. It will save. Okay, just I need a mole format of this molecule. I'm just copying it. And I want to calculate a ADMET properties for that molecule. Okay, so for the same molecule, this is a pre-ADMET tool where I can do ADME prediction. It will ask me from here, open. It will ask me a mole file. A mole file is copied from there and load it. See, this is the triclosan structure, which I got it. Just submit it. Maybe it will take a couple of minutes. Similarly, in the Swiss ADME, just you need to copy paste the, either you can draw molecule here or you can paste the SMILES notation and run it. It will give lots and lots of the ADMET properties. Okay, so from this, you can easily calculate all the ADMET properties. If you look at here also we got the pre-ADMET details. So what is what all these things, just if you want to need more details, just go to top here, ADMET prediction. Yeah, you can able to read all. It is the plasma protein binding, but the blood-brain barrier penetration, and also skin permeability and human intestinal absorption. So along with the differences, right? So with this, you can maybe if you have thousands of molecules, maybe it's difficult to calculate with these things. But if you have 100 molecules, maybe manually you can paste and you can do it here. Okay, these are the few very much important tools. Yeah. So databases. Yeah. If someone wants to go with the natural product databases, just go with the Coconut or Zinc. In Zinc, it will be a huge library, millions and millions. But in Coconut, only it will be having 400,000 natural product molecules with this. So directly you can download all the natural product and you can explore that. Or if you need a very small libraries, maybe 1500, 2000, 3000 molecules for the BPharm thesis or MPharm thesis. So directly you go to life chemicals databases, there they will be having various category of molecules like maybe antituberculous, anti-cancer drugs, or antibacterial. So maybe you can choose any one of the database and directly you can take it for the molecular dynamics or some other procedure. Okay, if you want to go ahead with the molecular docking, as I said that Autodoc is a freely open source, very good tool. It is one of the most accurate one. You can use Autodoc. It is initially you may feel little difficult for you to habituate to this software, but once you're good with us, why the reason I'm not explaining all the software here is time is one constraint. Another one is if just search YouTube. YouTube is like each and everything you'll get it in the YouTube tutorial. Okay, just explore it. Okay, another one is BioWare Discovery Studio Visualizer, just for preparation and visualization. It is very good, and Camera and PyMol also very good ones. Yeah. So next, coming to the end of these things, molecular dynamics. So in the molecular dynamics, GROMACS is one of the best software we have, and AMBER is another one. Next, this one. So this AMBER, these three are free softwares. This one is included in the Schrodinger software but academic purpose. It's the free one we have available is there. But all these softwares can be run only in the Linux machines, mostly in the Linux machines. So maybe you can install Ubuntu software by looking into the YouTube tutorials, just explore that. Maybe you can do that. Okay. Next, I'm going to share little my experience with how we have done some projects with me with different companies. I have done one project with the US-based company, another project with maybe New Zealand-based company. Just in the first project, what we have done is our main target is to build a natural product-based library. So from the various sources, I have downloaded different 500,000 plus molecules, and I have generated a library with that. And I have calculated all the parameters like number of aromatic bonds, aromatic rings, maybe hydrogen bond acceptors, donors, bond, like everything, all the properties were calculated, and we built an in-house database with that. And initially we started with that anti-aging targets, but after that a few days COVID got hit. So hence we moved our focus to the COVID, and we have generated, done with again 3 targets, spike and AC and purine targets. And for that, I have used a AWS cloud. Say 500,000 molecules, and on each target, I need to run 3 times. That means it is a 1.5 million molecules per against each single target. That equals to 3 targets. That will become a 45 lakhs of the molecules. So in the normal system, it's not possible. That is the reason myself I used AWS cloud, where we have chosen a 500 to 1,000 servers based upon the batch jobs. So that has done short for less than 5 hours of time. So, and it doesn't cost us much. It's only cost us less than $500, $400 to $500 for each run. So approximately with less than $2,000, we are done with the job. So based upon that, we have shortlisted some best ranked molecules. Still, it is going on. Maybe it takes time. I don't know. Okay. Another one is this one is we are working on drug repurposing. So here we have chosen the 500,000 natural products, but here we have chosen the molecules which were already approved by the FDA or European or Japanese medical agencies, and we want to choose those drugs as drugs for the COVID. So they have chosen two targets. I'm not revealing those two targets. Okay. So for one target, PDB protein is available. So straight away, we have gathered 1,000, like maybe 5,000 plus drugs from the various sources, and we have docked it. Another one is just that molecule, that target doesn't have a three-dimensional protein, the protein data bank. So we have used the homology modeling method to model that protein. Then we have generated this one, molecular docking. So another important thing is in the anti-aging target, we didn't have a specific binding site. So for that, we have used a blind docking. So what is meant by blind docking is we'll be using whole protein as our target. As I said that instead of choosing a small cavity, we are choosing the whole protein, means all complete 250 or 300 amino acids the protein. So our molecule will reach most of the cavities, and it tries to identify the best cavity for our target site. Yeah, with this, I'm just concluding my webinar on the CAD tools. Yeah. So first of all, I should thank CHIPS for giving me this opportunity, and also I should thank Subbarao sir for giving me generous opportunity, and principal sir. Yeah. So if you have any questions, I'm happy to answer. Maybe there is my presentation. Hope it makes some sense to each and everyone.
Yeah, it definitely made a lot of sense, first of all.
Yeah.
Just a minute, please, just a minute.
Maybe I'm a little faster, but I thought to cover so many things, but at the end, I couldn't be able to make it because it's been already 1.5 hours.
Yeah, but the content that you covered is completely worth, so we don't mind the time, but we had a few questions based upon the insights that you have given.
Okay.
But the first question is in the chat box. So the question was, is there any molecular weight range that limits for the selection of a protein or ligand preparation?
Yeah, when you're talking about the rule of 5, okay, molecular weight for the protein, there is no, for protein purpose, there is no molecular weight. So only the sequence, if sequence is, if for example, it is a 260 amino acid sequence. So it is a 260 amino acids, molecular weight is same for the n number of proteins, right? So there is no that kind of criteria. And coming to ligands, yeah, if you want to keep it as a rule of 5 criteria, rule of 5 should be less than molecular weight less than 500, acceptor should be less than 10, donor should be less than 5, and what is this? Lipophilicity should be less than 5. Yeah, but natural products, yeah, so we can able to violate some of the rules. So we usually, for example, Taxol, it won't fall under rule of 5. So rule of 5 is mainly we implement it for the synthetic drugs.
Okay. Okay. And the next question is, polar atoms are necessary for ligand preparations?
Polar atoms is necessary ligand preparation. Yeah, based upon your target cavity. See, if this cavity is really needed, a binding site is really needed some kind of polar groups, it's always good to keep a polar group into your ligand. If, for example, if it is a lipophilic acceptor kind of thing, so those kind of cases, you can keep it in polar group.
Okay, okay. And the next one is, will there be any changes, or can changes occur while we minimize the structure for the preparation of a ligand?
See, as I told earlier, there is no physical changes in this structure, but definitely there will be conformational changes. So the value minimum structure always it tries to get the least minimized energy, right? So if you as you're looking a perspective, yeah, structure will be looks different, but as a chemical perspective, there is no change.
Okay. And the next one is, can all the force fields be determined by using a single docking software, or there are different softwares for that?
What is meant by all four fields? All four fields, right? Maybe they were considering like docking force or hydrogen bond interactions and the docking score. Yeah, all three should be determined by the only single docking software.
Okay. Okay. And can probiotics be possibly designed using any computer-aided drug design?
See, the probiotic itself, there is no specific, I don't think there is a specific structure for the probiotic. It might be like composition of multiple things. So if you want to do any kind of computational study, we should have a clear structure, either protein or ligand with us. That is a single structure.
Okay.
Then only we can able to do it. But there were some studies in the molecular dynamic point of view. People are maybe trying in the complex kind of things. So maybe in the molecular dynamics, maybe we can try it.
Okay. Okay. And then another question that we have is the reproducibility of these softwares. So today, if I have a structure today and I start working on it, and after a few weeks, I submit this and other person tries to do the same thing in another software, will there be reproducibility?
Yeah, changing for the softwares, according to the theoretically, we should get the same reproducibility. See here, the rank numbered one molecule in the different software also should be the rank numbered one, but practically we will not get 100% reproducibility. Maybe we'll be getting 70 to 80%. Maybe here is the top number one molecule. There it might sit as a top 10 molecule.
Okay. And as you said that these softwares, which are paid softwares, they can be afforded by the companies, but not when you are doing a research in an educational institute. So will the accuracy that you get with these softwares that you have mentioned, the same accuracy and precision can also be obtained, can be maintained with the paid softwares?
See, for example, if you take a Schrodinger, there just assume that you will be getting 90% accuracy. Okay. But if you're doing it with the Autodoc, just assume that you'll be getting 70 to 80% accuracy. So there is cost matters. There is a lot of efforts matters for the precision might be a little different because those people are investing lots of money into developing a software. But these are the educational. This Autodoc is like discovered by the Scripps Institute. It's USA. So they may not have that much of funding to develop this kind of software. But yeah, according to studies, they have the success stories. In Autodoc also they have the success stories, and there they have the failures. Here also we have the failures. So as an educational point of view, so it's the better, very good starting point for us.
Okay. So these softwares will definitely give us the precision with which we can carry our research forward.
Yes, right.
Okay. So that's it. So these are the questions that we have for you. And I should wholeheartedly say that not only the information that you have given us, but the demonstrations that you gave using different point of view, like the protein bank, etc., were very, very informative. And I really wish that the students, especially the research students who are working on this, will definitely get benefited out of yours. And we really thank you once again on behalf of Chabrol Hunma Institute of Pharmaceutical Sciences for being the speaker for today's session and putting forward all your expertise, all your knowledge into this. We once again thank you so much. And with this, I would like to say to all the participants that we came to the end of this webinar, and I would like to conclude this webinar by proposing a very small vote of thanks. Well, at the outset, I would like to thank the entire management of Chebroil Halmaya Institute of Pharmaceutical Sciences for giving us the go-ahead to carry this series of webinars so that we can definitely bring a lot of information, a lot of advancements in the science that we are a daily part of. So thanks to the management. Next, I wholeheartedly thank the principal, Dr. S. Vidyadhara, for giving us the free hand to contact the persons, contact the different expertise, and bringing them on this platform so that they can deliver their expertise on this platform. And I also congratulate the Department of Pharmaceutical Chemistry for making this webinar possible. And last but not the least, I thank all the participants for your active participation. And I really hope that we'll meet once again on this similar platform for further expertise. Thank you. We'll end this webinar with this note. Thank you once again.
Thank you all.